Welcome back to the deep dive. I want to start today with a mental image. I want you to imagine something that has no brain, no stomach, no lungs, and no muscles.
No moving parts at all, really.
Right. It cannot move on its own power. It cannot eat. It cannot breathe. If you leave it on a table, it is, for all intents and purposes, a rock. A very complex rock, but a rock nonetheless.
It sounds a bit like a riddle when you put it that way.
It does. But this rock is responsible for wiping out entire civilizations, shutting down the global economy for years at a time, and killing more humans throughout history than all wars combined. We are talking, of course, about the virus.
The ultimate minimalist of the biological world.
Exactly. And today we are doing something different. We aren't just reading a Wikipedia list of bugs. We are going deep into the architecture of the enemy. We are basing this entire conversation on Murray's Medical Microbiology, specifically Chapter 36, while supplementing a bit from Jowett's Chapter 29.
Which is the fundamental chapter. It's the one that sets the rules for literally everything else in virology.
Right. And I know for a lot of medical students, this chapter can feel dry. It's taxonomy, it's structure, it's, memorize this shape or that size. But my goal today, our mission for this deep dive, is to prove that if you actually understand the engineering of a virus, you don't have to memorize the lists. You can predict them.
That is the key, predictive modeling. If you know how the machine is built, you know how it breaks, and you know how to break it therapeutically.
So let's start with the existential question. What is a virus? Murray gives a very specific history here that I think is fascinating.
He starts with the historical term filterable agents, which takes us all the way back to the late 1800s.
Honestly, it sounds like a Cold War spy novel title, The Filterable Agent.
It really does, but it was a very practical lab reality at the time. Back then, the germ theory was just taking off. We finally knew bacteria caused disease, and we had these porcelain filters, Chamberlain filters, that had microscopic pores. If you had a fluid full of bacteria and you pushed it through this filter, the bacteria would get stuck. The fluid coming out the other side was completely sterile.
So if you injected that filtered fluid into a healthy animal, nothing happened. You'd essentially purify the water.
Exactly. Unless, and this is where it gets weird, unless it was something else entirely. They found that with certain diseases like tobacco mosaic disease in plants or foot and mouth disease in cattle, the fluid that passed through the filter was still highly infectious. The agent was so small, it slipped right through the net, hence filterable.
So initially, the definition was literally just invisible small thing.
Right. It was defined by what it wasn't. It wasn't a bacteria. But obviously, we have a much better definition now. Murray defines viruses as obligate intracellular parasites.
I want to stop on that phrase for a second. Obligate intracellular parasite. Because I feel like we use the word parasite incredibly loosely in everyday language. Like, my cat is a parasite. A tapeworm is a parasite.
Well, don't let your cat hear that. But biologically speaking, there is a massive distinction here. A tapeworm living in your gut is a parasite, yes. It steals your food, but it has its own metabolism. It takes in nutrients, it processes energy, it builds its own cells. It's an independent organism freeloading on your real estate.
And a virus?
A virus has none of that. It has absolutely no metabolism. It cannot generate ATP for energy. It has no ribosomes to make proteins. This is the obligate part. It is not just that it likes being inside a cell. It is that outside of a cell, it ceases to function biologically. It is just complex chemistry waiting for a spark.
So it's like a blueprint floating in the wind looking for a contractor to build the house.
That is the perfect analogy. And this leads to arguably the most important distinction Murray makes between bacteria and viruses, the fundamental difference between division and assembly.
Binary fission versus the Lego set.
Precisely. If you look at a bacterium, say E. coli, it grows. It physically gets bigger. It replicates its DNA. It forms a wall down the middle. 1 becomes 2, 2 become 4. It is a continuous process of cellular division.
But you never see a baby virus growing into a big virus.
Never. Viruses are manufactured like cars on an assembly line. The infected host cell makes the parts separately. It turns out 1,000 copies of the genome over here. It turns out 10,000 copies of the protein shell over there. It makes the enzymes. And then usually at the very end of the cycle, all those parts come together, snap, 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 and they literally assemble into the final infectious particle.
Which Murray officially calls the virion.
Right. The virion is the physical particle. The virus is the broader biological entity.
This assembly concept is huge because it explains why viruses are so prone to errors, which we'll definitely get to. But first, we have to talk about the names. Because if you open a microbiology textbook, the names seem to have been chosen by a committee of drunk historians.
It is a bit of a graveyard of old scientific ideas, yes.
I mean, we have names based on size, on shape, on where they were found. Give us a tour of the concept cars of viral naming.
We'll take pacornavirus. That one is actually quite helpful. It's a compound word. Pico means very small. RNA is the genome. Pico-RNA virus, small RNA virus.
Okay, I can respect that. It's functional. It tells you exactly what you're dealing with.
Then you have toga virus.
Like the Roman toga.
Exactly like the Roman toga. It refers to the membrane envelope that wraps around the virus like a cloak. It's a virus wearing a toga.
That's fantastic. What about adenovirus?
Named after the anatomical site where it was first isolated, the human adenoids.
And my personal favorite, the reovirus.
Ah, the orphan.
Explain that one to the listeners. It sounds so sad.
It stands for respiratory enteric orphan virus, REO. When researchers first discovered it, they found it in the respiratory tract and the enteric tract, meaning the gut, but they couldn't pin it to a specific disease. It was a virus without an illness, an orphan looking for a home.
Poor little guy. Though I assume it's not actually harmless.
Oh, not at all. Rotavirus is in that family, and it's a major killer via pediatric diarrhea worldwide. The orphan grew up to be quite a delinquent.
But the main point Murray makes is that while these historical names, retrovirus, togavirus, bunyavirus, stick around in the literature, the modern medical way to classify them isn't by a catchy name, it's by hardware.
Correct. We have moved away from where did we find it to what is it actually made of. To classify a virus today, you ask 3 definitive questions. 1, how big is it? 2, what is the shape of the shell? And 3, most importantly, what is the genome made of?
Okay, so let's do a deep dive into that hardware. Section 2 of our roadmap, the physical structure.
Think of the virus as a package you get in the mail. You have the payload, which is the genome, and the container, which is the capsid and sometimes an envelope.
Let's start with the payload, the genome. Murray lays down a hard, unbreakable rule here.
The rule is simple. DNA or RNA, never both.
Which is unique, right? Because I have both.
You, me, your cat, the bacteria in your gut, the fungi on your shower curtain, we all use both. We keep our master files locked away in DNA, and we use RNA as the temporary messenger notes to get work done on the factory floor. Viruses pick a lane, they commit to one or the other.
And that choice DNA versus RNA isn't just a trivial trivia fact. It dictates literally everything about how they behave clinically.
Everything. Let's look at the DNA viruses first. DNA is a chemically stable molecule. It's the ultimate archivist. If you leave DNA at a crime scene, forensics can sequence it 20 years later.
So DNA viruses are stable.
Very stable. Furthermore, they have proofreading enzymes. When they copy themselves, they check their work. They don't change much over time. Smallpox today is fundamentally the same as smallpox 1,000 years ago. That genetic stability is exactly why we could eradicate it with a single vaccine. The target didn't move.
Contrast that with the RNA world.
RNA is the Wild West. Chemically, it's labile. It falls apart easily if you just look at it wrong. Biologically, the enzymes that copy RNA are incredibly sloppy. They make typos constantly, and they have absolutely no proofreading ability to fix them.
So they mutate constantly.
They mutate rapidly. This is the concept of genetic drift. It's why you need a new flu shot every single year. The virus you fought last year has drifted enough that your immune system's antibodies simply don't recognize it this year.
So DNA implies stability and reliable vaccine targets. RNA implies adaptability, rapid evolution, and chaos.
Exactly. That is a foundational mental model you need for the boards.
Now, within that chaotic RNA world, there's a concept that Murray stresses heavily, and honestly, it's one that always trips students up. The concept of sense. Positive sense versus negative sense RNA.
This is immensely high yield. If you are prepping for a medical exam, you need to understand this deeply.
Let's break it down with an analogy.
Okay. Think of the human host cell's ribosome, our protein factory, as a construction crew that only speaks one language. Let's say English. English is their native language. In molecular biology, that native language is messenger RNA, or mRNA.
So if I hand the ribosome a strip of mRNA, it knows exactly what to do.
It immediately reads it and starts building proteins. No translation needed.
Okay, so what is a positive sense RNA virus?
A positive sense genome is written in perfect English.
Yeah.
It structurally looks exactly like host mRNA.
So if a poliovirus, which is positive sense, injects its genome into my cell,
The construction crew picks it up and says, hey, instructions, and they start building viral proteins immediately. They don't know they're building a biological weapon. They just follow the instructions in front of them.
so the positive sense virus is ready to rock the exact moment it enters the cytoplasm.
Correct. Now, what about negative sense?
Right. How does the opposite work?
Negative sense is like the photographic negative of the image. Or, sticking to our construction analogy, it's text written in mirror image, or a foreign language the Cree doesn't speak.
So if a negative sense virus, like say rabies or measles, injects its genome into my cell, and that's all it does,
What happened?
absolutely nothing. If human ribosome looks at it, can't read a word of it, and ignores it, the infection completely fails.
So the virus has a massive problem upon entry.
A huge problem. It has to convert that negative sense strand into a positive sense strand before it can do anything at all. But the host cell doesn't have a mirror reader enzyme for RNA to RNA transcription. We only transcribe DNA to RNA.
So the virus has to bring its own mirror.
Exactly. This is the golden rule of negative sense viruses. They must carry a fully formed viral polymerase enzyme packed physically inside the viral capsid. They travel with their own translator.
That is a critical distinction. Positive sense.
Very-
The genome itself is the infectious message. Negative sense. The genome is just a template, and you must bring the physical tool to read it.
Precisely. If you chemically purify just the RNA of a positive sense virus and inject it into a cell, you get an infection. If you purify the RNA of a negative sense virus and inject it, nothing happens, because you left the polymerase tool behind.
That's the exact kind of mechanism-based reasoning that saves you from rote memorization.
There is one more genomic feature we need to mention before we move on to the shell: segmentation.
Right. The chapter book versus the scroll.
Most viruses have a single continuous long strand of genetic material, a scroll, but some, specifically influenza from the orthomyxoviruses and reoviruses, have genomes chopped into distinct, separate pieces.
Like individual volumes of an encyclopedia.
Right. Influenza famously has 8 segments, 8 separate physical pieces of RNA floating inside the capsid.
And this isn't just a quirky filing system choice. This has massive world-altering implications for pandemics, which we'll touch on in the genetics section. But I want you listeners to file that away. Segmented genomes allow for shuffling.
The marble jar effect, we will definitely come back to that.
Okay, so that's the payload. Now let's look at the container itself, the capsid.
The protective protein shell.
Murray describes 2 main architectural shapes here. The icosahedral, which is essentially the soccer ball, and the helical, which is the rod.
Let's look at the soccer ball first. Why does nature love this specific shape? Why are so many viruses, polio, herpes, adenovirus, shaped precisely like a 20-sided die?
I'm assuming it's not because nature loves playing Dungeons and Dragons?
It's not. It's entirely about genetic economy. Remember, the virus is a minimalist. It has a tiny, tiny genome. It doesn't have the storage space for complex architectural blueprints requiring 50 different types of building materials. It wants to build a secure shell using the absolute smallest possible number of unique parts.
So it uses basically one type of brick.
Exactly. If you take one simple protein subunit and repeat it over and over, the icosahedron is the most efficient mathematical way to enclose a three-dimensional space. It is incredibly thermodynamically stable. It essentially solves itself. You could put the individual viral proteins in a beaker of water, shake it, and they will naturally snap together into these perfect spheres.
Self-assembly. Magic.
It's elegant chemistry.
And the rod. The helical shape.
That's where the protein subunits bind directly to the genetic coil itself, spiraling around it like a rubber coating on a metal spring. Now, here is a rule from Murray that is absolute gold for human virology.
Let's hear it.
In human viruses, if a virus has a helical nucleocapsid, it is always enveloped.
Always. No exception.
Always. You can have naked helical viruses in plants, like the tobacco mosaic virus we mentioned earlier, but not in humans. If it looks like a rod under the electron microscope, it is wearing a lipid coat. Think influenza, rabies, Ebola, they are all enveloped rods.
Okay, you just perfectly set up the most clinically relevant structural feature of all. The distinction between the naked capsid virus and the enveloped virus.
Yes, this is the tank versus bubble analogy. And honestly, if you take only one single concept from this deep dive to the hospital wards, make it this one.
Let's start with the tank, the naked virus.
A naked virus is just the genome and that tough protein soccer ball we talked about. There is no outer lipid layer, just hard protein.
And protein is tough.
Protein is very tough. Think of a thick wool sweater. You can dry it out, it's fine. You can get a little acidic, it's fine. Naked viruses are environmentally stable, they resist heat, they resist drying out on surfaces, and most importantly for us, they resist detergents and acid.
Acid, like human stomach acid.
Exactly. Because they can survive the harsh hydrochloric acid of the stomach and the bile salts of the intestines, naked viruses can be transmitted via the fecal-oral route.
Which is the medical way of saying poop to mouth.
It is. But think about the clinical picture here. What viruses sweep through daycares or rip through cruise ships, causing explosive diarrhea and vomiting? Norovirus, rotavirus,
Naked viruses.
or polio, hepatitis A. These are all viruses that are shed in massive amounts in stool, survive in the sewage or on a bathroom doorknob for days, survive on your hands, get swallowed, survive the stomach acid, and successfully infect the lower gut. They are microscopic tanks.
Got it. Now contrast that with the bubble, the enveloped virus.
An enveloped virus has literally stolen a piece of the host cell's membrane to wrap around itself as it leaves. This envelope is made of lipids, fats.
And what happens to a tiny fat bubble when it sits on a dry table?
It pops, it degrades. Lipid membranes desperately need moisture to maintain their structural integrity. What happens to grease when you hit it with simple soap or detergent?
It dissolves immediately. That's how we wash our dishes.
Exactly. So enveloped viruses are total wimps in the external environment. They are labile. They cannot survive drying out. They generally cannot survive the stomach acid. And they're completely destroyed by simple hand soap or alcohol sanitizer.
So how on earth do they transmit from person to person?
They need to stay wet. They need a direct, moist line of transit from one warm host to another.
So, respiratory droplets.
Yes. Sneeze directly to the face, yes. Influenza, COVID-19, RSV,
Blood to blood.
HIV, hepatitis B and C,
Sexual transmission.
herpes, HIV.
So if I'm a student and I get a test question about a virus transmitted by sharing a dirty needle or through sexual contact, I should immediately be thinking, enveloped.
Highly likely, yes. And if the clinical vignette is about a virus surviving in a public swimming pool or on a plastic toy in a nursery, you think naked.
There are exceptions, right? Biology always has exceptions.
Of course. Pox virus is enveloped, but it has a very complex, multi-layered, brick-like structure that makes it much tougher than a standard bubble. But as a general heuristic, the tank versus bubble rule holds up brilliantly.
One last structural component before we get into the life cycle. The keys.
The viral attachment proteins, or VAPs.
Because a virus can't just float into a cell by magic. It needs to physically dock.
It needs a very specific handshake. On the surface of every single virus, there are molecular structures designed to fit perfectly into specific receptors on the human cell.
If you are a naked virus, these keys are just built into the capsid itself.
Right, they form ridges or canyons right on the surface of the protein's soccer ball.
But if you are an enveloped virus, they are spikes poking through the fat bubble.
Like the famous spike protein on coronavirus or the hemagglutinin on influenza. These VAPs are the primary targets for our immune system's neutralizing antibodies. If your body coats the key with an antibody, it gums up the works. The virus physically can't unlock the door, and the infection is stopped before it starts.
Okay, so we have our fully built virus. We know if it's a tank or a bubble. We know if it's carrying a positive or negative blueprint. Now, the invasion begins. Section 3 of our outline, the replication cycle.
Murray refers to this as the host-pathogen chess match.
I love that framing. And it happens in distinct ordered phases. Murray lists them out: attachment, entry, uncoating, replication, assembly, and release. But before we go step by step, he introduces a fascinating concept called the eclipse period.
The eclipse period is almost philosophical.
Why do you say that?
Because it marks the exact moment the virus functionally dies to be reborn. When a virus enters a cell, it has to uncoat. It has to break down its protective shell to release the genome into the cell. At that moment, if you looked under an electron microscope, you wouldn't see a virus. If you ground up the cell and tried to infect another cell, you wouldn't find an infectious particle.
It is totally disassembled.
It is gone into eclipse. It literally ceases to exist as a physical entity until the new parts are built and fully assembled hours later.
Let's walk through the steps to get there.
Yeah.
Step 1, attachment, the lock and key.
This step single-handedly determines tropism. Tropism is just a fancy virology word for tissue preference. Why does hepatitis destroy the liver but completely ignore the brain? Why does HIV obliterate T cells but leave muscle cells alone?
Because the viral keys only fit those specific cellular locks.
Exactly. HIV's key, which is a glycoprotein called GP120, perfectly fits the CD4 lock. CD4 is primarily on helper T cells and macrophages. So the virus ignores 99% of your body and mercilessly hunts down the immune system.
Or Epstein-Barr virus, EBV.
Right, EBV binds to the C3 receptor, also known as CR2, strictly on human B cells. It's a very specific targeted address system.
Step 2, entry, We're attached. Now, how do we get inside?
Well, if you are a naked virus, you usually trick the cell into eating you. It's called receptor-mediated endocytosis or viropexis. The cell membrane wraps around the virus and pulls it inside in a little sac.
The classic Trojan horse maneuver.
Exactly. But if you are an enveloped virus, you have a much slicker option, membrane fusion.
This is what HIV does, right?
Yes. The viral envelope is made of the exact same lipid material as the host cell membrane. Oil and oil, they merge. The virus touches the surface, the two lipid membranes melt seamlessly together, and the viral capsid just slides right into the cytoplasm.
But some enveloped viruses, like our old friend influenza, do a combination of both.
Influenza is incredibly sneaky. It gets eaten via endocytosis first, so it's sitting inside this cellular bubble called an endosome, deep inside the cell. The cell, trying to defend itself and digest the invader, starts pumping acid into the bubble.
The cell thinks it's dissolving the virus.
But for influenza, the acid is the exact trigger it was waiting for. The sudden drop in pH causes a massive physical shape change in the viral spike protein. The hemagglutinin snaps open like a microscopic switchblade, grabs the lipid wall of the endosome bubble, and forces fusion right there from the inside.
So the cell's own defense mechanism, the acid, is actually the key the virus relies on to escape and infect.
Evolution is terrifyingly clever.
Step 3 is uncoating, which we just talked about. The eclipse begins, the shell comes off, the genome is completely free.
Which brings us to step 4, macromolecular synthesis. The factory floor.
This is exactly where students get lost in the weeds. They try to memorize, okay, this one goes to the nucleus, this one stays in the cytoplasm, this one goes to the Golgi. Is there a general rule to anchor this?
There are 2 foundational rules, the nucleus rule for DNA viruses and the cytoplasm rule for RNA viruses.
Let's do the DNA rule first.
Think about it logically. If you are a DNA virus, you need the cellular tools to read and copy DNA. Where does the human cell keep all of those heavy-duty tools?
Locked securely in the nucleus. That's the master library.
So almost all DNA viruses enter the cell and immediately commute straight to the nucleus. They hijack the host's DNA-dependent RNA polymerase to make their mRNA.
Is there an exception? Because there's always an exception.
There is always a big exception, and in this case, it literally is big. Pox virus.
Like smallpox.
Yes. Pox viruses are physically huge. They are so big, they are almost bordering on bacteria size. Because they are so large, they can carry their own massive factory with them. They have their own DNA polymerase, their own RNA polymerase, everything. They don't need the host library at all. They just set up shop right in the cytoplasm and do everything themselves.
So poxvirus is basically the sovereign, self-sufficient nation of DNA viruses.
Correct. DNA equals nucleus except pox, which is cytoplasm.
Now the RNA viruses.
The human cell doesn't really replicate RNA from an RNA template. You don't do that. So the nucleus is largely useless for an RNA virus. Therefore, the vast majority of RNA viruses stay out in the cytoplasm and build their own replication complexes right there.
Any exceptions here?
Influenza, the orthemic subviruses, and retroviruses like HIV, they go to the nucleus.
Wait, why influenza? I thought we established it was an RNA virus.
It is, but influenza is a pirate. To make its viral mRNA readable by the human ribosome, it needs a special chemical hat on the end of the RNA strand called a 5' cap. It doesn't have the enzymes to make one, so it travels into the host nucleus, finds the host's freshly minted mRNA, and literally cuts the heads off.
Cap snatching.
It's technically called cap snatching. It steals the caps to wear them itself so it looks legitimate to the ribosomes. That process can only happen in the nucleus.
And retroviruses, why do they go to the nucleus?
Well, retroviruses are the ultimate rule breakers of biology. They enter as RNA, but they carry an enzyme called reverse-transcriptase. They use it to turn their RNA genome backwards into DNA.
You mean retro?
Exactly. Once they are converted into DNA, they travel into the nucleus and use another enzyme called integrase to physically splice themselves right into your human chromosome.
And once they're integrated into my DNA?
They are biologically a part of you. Every single time that host cell divides, it flawlessly copies the viral code along with your own. This is exactly why HIV is a lifelong chronic infection. You can't just scrub the viral blueprint out of the library archives once it's written into the books.
Okay, so the factory has run its course. We have fresh genomes. We have newly minted proteins. Step 5 is assembly.
Which we touched on. Thermodynamic crystallization. The puzzle pieces naturally snap together into mature variants.
And finally, step 6. Release. The exit strategy.
This goes back to our tank and bubble analogy. If you are a tank, a naked virus, your exit strategy is usually quite brutal. Lysis. You multiply and fill the cell until it literally bursts open. The host cell dies dramatically and thousands of viruses are released into the tissue at once.
If you are a bubble, an enveloped virus,
You are much stealthier. Budding. You push gently against the inside of the host cell membrane. It bulges out, wraps around your capsid, and slowly pinches off. You steal your lipid coat on the way out the door.
does this budding process kill the cell?
Not immediately, no. In fact, the cell can remain alive, acting as a zombie factory, chronically shedding virus for weeks or even years. This is the mechanism behind persistent infections like hepatitis B or C.
I want to mention one very specific, highly testable drug target here regarding release, neuraminidase.
Ah, the Velcro problem.
Explain the Velcro problem to us.
So, influenza successfully buds out of the respiratory cell, but its surface spikes, the hemagglutinin, naturally stick to sialic acid receptors on the outside of the cell surface. As the newly formed virus tries to float away, it gets tightly tethered to the cell it just left.
Like getting your belt loop securely caught on the door handle as you try to walk out of a room.
Exactly. So influenza carries a pair of molecular scissors called neuraminidase. Its only job is to cut the sialic acid tether so the new virus can fly free and infect the next cell.
And this is exactly where Tamiflu works.
Yes. The generic name is oseltamivir, and it is a neuraminidase inhibitor. It aggressively gums up the scissors. So what happens? The virus replicates perfectly, it buds out perfectly, but it can't leave. It stays completely stuck to the dying host cell, and the infection is halted in its tracks.
That is such a deeply satisfying mechanism to understand.
It is. It makes the pharmacology so much easier to remember.
So we've built the virus, we've replicated it, we've released it. Now, briefly, section 4 of our outline, viral genetics, or how the enemy evolves.
We already touched on the fundamental mutation rates. DNA equals highly stable. RNA equals sloppy high mutation rate.
But there is a specific genetic phenomenon called reassortment that we absolutely need to clarify, because this is the true pandemic generator.
This goes right back to the segmented genome concept we discussed, the marble jar.
So imagine a virus genome isn't a long, continuous scroll, but rather a jar containing 8 distinct marbles.
Right. Say you have an avian flu virus, a bird flu. Let's say its 8 genome segments are red marbles. And you have a common seasonal human flu virus. Its 8 segments are blue marbles. Now, a pig on a farm, and pigs are unique because their cells have receptors that can catch both human and avian strains. This pig gets infected by both viruses at the exact same time in the exact same cell.
The pig's respiratory cell is now essentially full of a mix of red and blue marbles.
During the assembly phase, when the new virions are being built, the capsid just grabs 8 marbles to fill the jar. It doesn't check the color. So you might get a progeny virus that accidentally packs 5 blue human marbles and 3 red avian marbles.
And if one of those red avian marbles happens to code for a brand new surface hemagglutinin spike that the human population has never, ever seen before.
You have a nightmare scenario. You have a virus that replicates beautifully and transmits easily in humans thanks to the blue human genes, but looks completely alien to the human immune system thanks to the red bird genes. That is called antigenic shift, and that is exactly how we get massive, deadly global pandemics.
And crucially for the exams, this only happens in segmented viruses.
Correct. Primarily influenza and reovirus. Polio cannot do this. Herpes cannot do this. They have single continuous scrolls. You can't reassort a single scroll.
What about recombination? How's that different?
Recombination can happen in viruses with single long genomes, mostly DNA viruses. That's more like traditional chromosomal crossing over. 2 homologous strands line up, break, and physically swap a chunk of DNA. It happens, but it's a much slower evolutionary process compared to the sudden, massive shuffle of reassortment.
Finally, section 5. Clinical and therapeutic connections. We've been weaving these in throughout the deep dive, but let's quickly summarize the overall antiviral logic based on what we've learned.
The entire logic of virology pharmacology is attack the unique machinery, find the step the virus does that the human cell doesn't do.
For instance, entry inhibitors.
Right. Block the receptor handshake. Maraviroc for HIV physically blocks the CCR5 co-receptor on the human cell. The key can't enter the lock.
Uncoating inhibitors.
Block the acid-triggered escape. Amantadine was used to block the M2 ion channel in influenza, stopping the acid from entering the viral core. Though, as we noted with RNA viruses, resistance mutates quickly, so we rarely use it now.
Polymerase inhibitors. This seems to be the really big one.
It is the biggest class. Acyclovir for herpes, AZT for HIV, sofosbuvir for hepatitis C. The mechanism is brilliant. You feed the patient a fake, defective nucleotide building block. The viral polymerase is sloppy and fast, so it accidentally eats the poison pill and halts replication. The human cellular polymerase is smart and has proofreading, so it ignores the fake building block.
And lastly, release inhibitors, which we covered.
Exactly, the neuraminidase inhibitors like Tamiflu keeping the virus tethered to the cell.
It's amazing how much of clinical pharmacology is just directly exploiting the structural differences we just spent an hour discussing.
That's the whole game. Find the part of the microscopic machine that humans don't naturally possess and firmly jam a molecular wrench in it.
Listen, this has been a whirlwind tour of virology. I feel like I can actually visualize these little machines working now.
That's the entire goal. Don't memorize abstract lists. Visualize the tank surviving the stomach. Visualize the fragile bubble popping on a dry table. Visualize the assembly line factory floor.
Are you ready to test the listeners?
I'm ready. Let's do it.
We are going to do 5 rapid-fire, single best answer style questions to consolidate this. I'll ask the question, I'll give you a brief pause to think, and then our expert will reveal the answer and the mechanism.
Remember to use the mental models we just built.
Question 1. A 5-year-old child presents to the clinic with severe vomiting and watery diarrhea. The causative virus is isolated and shown in the lab to be highly resistant to degradation by detergents and low pH environments. Is this virus likely naked or enveloped? And based on that, what is the primary mode of transmission? I'll give you a second to think about that.
Okay, the virus is naked, and the answer is fecal-oral transmission. Recall the tank versus bubble model. Enveloped viruses, the bubbles, are quickly destroyed by stomach acid and detergents. Naked viruses, the tanks, easily resist the gastrointestinal environment. Therefore, profound GI symptoms plus chemical resistance strongly points to a naked capsid virus like norovirus or rotavirus. If you miss that, review the naked versus enveloped section in Murray.
Excellent. Question 2. You are an infectious disease researcher analyzing a novel virus. You determine it contains a single-stranded, negative-sense RNA genome. What specific protein enzyme must be physically present inside the infectious virion particle for an infection to successfully initiate? Take a moment.
The answer is an RNA-dependent RNA polymerase. Remember the language analogy. Negative-sense RNA is a completely foreign language to the host ribosome. The host cannot read it. Therefore, the virus cannot rely on the host to translate anything initially. It absolutely must bring its own physical enzyme tool to transcribe the negative strand into a readable positive mRNA strand immediately upon entry into the cytoplasm.
Question 3. A large DNA virus is observed under electron microscopy, replicating entirely within the cytoplasm of the host cell, producing large visible inclusion bodies. Which specific virus family does this most likely belong to? Think about the rules.
The answer is poxviridae, or poxvirus. The general central dogma rule for virology is that DNA viruses replicate in the host nucleus to use the host's enzymes. Poxvirus is the giant, notable exception to this rule. It is so exceptionally large and complex that it encodes and carries all of its own necessary transcriptional machinery, allowing it to bypass the nucleus entirely and replicate out in the cytoplasm.
Question 4. Which of the following structural features is always present in human-infecting viruses that possess a helical nucleocapsid symmetry?
The answer is an envelope. This is a strict, unbreakable morphology rule for the boards. In human virology, there are absolutely no naked helical viruses. If the viral capsid is shaped like a rod or a helix, such as influenza, measles, or rabies, it is universally surrounded by a lipid envelope.
Last one. Question 5. Devastating pandemic influenza strains are most often the direct result of the exchange of genomic segments between human and animal viral strains within a co-infected host. What is this specific genetic shuffling process called?
The answer is reassortment. This is our marble jar concept. It is fundamentally distinct from recombination. Reassortment involves the wholesale shuffling of entire intact gene segments, and crucially, it can only occur in viral families that possess segmented genomes, primarily the orthomyxoviridae like influenza and reoviridae like rotavirus.
If you nailed those 5 questions, you are officially ready for the virology awards. If not, don't panic. Go back and actively read Murray chapter 36. Look at the specific diagrams of the viral replication cycles in the text.
Seeing the visual layout of the factory floor really helps lock these concepts into your long-term memory.
Absolutely. Now, before we wrap up, I want to leave you with a final thought to mull over. We talked extensively about how viruses are obligate parasites, entirely dependent on our cells to exist. But consider this. About 8% of your entire human DNA genome is made up of ancient retroviruses that integrated into our ancestors' germline millions of years ago and just stayed there. Some of those viral genes are now essential for human placental development. So who is really hijacking whom at this point in our evolutionary history?
That is a brilliant and slightly unsettling thought to end on.
Thanks for joining us on this deep dive. Keep learning, stay curious, and honestly, wash your hands with soap and water, especially if there are naked viruses going around. Hand sanitizer won't save you from the tanks.
Absolutely true. See you next time.